because before I want to introduce our speakers, I just want to take this opportunity to thank the fantastic group of individuals who helped us put this part of the program together. They are Birgitte Henriques Normark at the Karolinska Institute, Johan Bengtsson Palmer of the University of Gothenburg, Pontus Nordenfelt at Lund University, and Sanna Koskiniemi, Uppsala University. So thank you all very much for your ideas and making your network available for putting this session together. Which brings me to the session. What I would like to do with this session, and what we discussed we would do, is have three speakers who give us an idea of what we think the flavor of data-driven infection biology is, and but that we allow some time to have a joint discussion in the end to explore this further. And I would therefore ask the speakers to limit themselves to 20 or 25 minutes at the maximum, and then if we can take one or two technical questions from the chat at the end of each talk, but I would really like to allow some time for overarching questions as to what data-driven infection research is and should be in future, so that we can have that discussion in the last 10 minutes or so at the end of this session. The first person I would like to introduce as a speaker is Dr. Laura Carroll. She is currently finishing a postdoc at the EMBL with Georg Seller's lab at EMBL Heidelberg. She completed her PhD as recently as 2019 at Cornell with Martin Wiedmann. And she labeled herself on one occasion recently as a computational microbiologist and bioinformatician who studies the evolution and transmission dynamics of pathogens. And that reflects a fairly diverse approach that she takes, and I anticipate she will give us a flavor of these broad range of approaches that she's taking today. And I'm happy to say that Laura will join the DDLS program as one of the first fellows that have been recruited, and she will join Umeå University from August. So Laura, please take it away. That looks good.
Good. I'm always like paranoid y'all can see my weird notes to myself. Hey everyone, my name is Laura Carroll. Thank you so much for the awesome introduction. I'm a postdoctoral fellow currently working at EMBL in Heidelberg. I'm a computational biologist, and today I'm going to talk to you about how we can leverage genomic data to track and identify emerging bacterial pathogens. So as I'm sure all of you are acutely aware, living through a global pandemic and whatnot, emerging infectious diseases, including newly, re-emerging, and deliberately or accidentally emerging diseases, pose a serious threat to global public health. Probably could skip that slide. But to that end, massive amounts of whole genome sequencing data from pathogens has and continues to be generated, and it's likely that efforts to sequence them aren't slowing down anytime soon. However, not only do we have this continuously growing repertoire of genomic data derived from pathogens, but we also have this increasing stream of metadata associated with these pathogens. So for example, like the geographic origins of strains, we might have information regarding the host or environmental source associated with the pathogen. Maybe we're lucky and we have phenotypic data, such as whether strains are resistant to antimicrobials or not, or maybe if they produce some sort of toxin or not. And my personal favorite to work with, temporal data. So maybe we have the year or date that a given strain was isolated. And using all of this publicly available data and metadata, there's a lot of really cool biological and epidemiological questions that we can ask about pathogens. So for example, we may be interested in the geographic origin or the host or environmental origin of a pathogen, or we might be interested in figuring out which time frame a given pathogen became a problem in the human population. Or maybe we're interested in what exactly makes this particular pathogen so special or so problematic. Maybe it has some sort of novel virulence factor. Maybe it's resistant to treatment, for example, with antibiotics. Maybe it's more or less transmissible, or maybe it causes some sort of novel or unexpected symptoms. So in my research, I attempt to answer all of these sorts of questions using in silico approaches. And because I'm a little more on the method side, especially lately, I work with a ton of different organisms and systems, so I had a hard time figuring out what to talk about today. But I decided on a couple of recent projects that I've been working on, which showcase how you can use these sorts of data and metadata to answer these sorts of questions. And I'll start with this super understudied emerging pathogen called Bacillus Series. So Bacillus Series is a gram-positive spore-forming organism that's widespread throughout the environment. As a pathogen, B. Series has been associated with numerous forms of illness, including severe or fatal non-gastrointestinal infections, but usually it's seen as a foodborne pathogen that's estimated to be responsible for more than 256,000 foodborne illnesses globally. But this number is likely severely underestimated due to the fact that foodborne illness caused by B. Series is typically very mild. It rarely lasts longer than 24 hours, and more often than not, cases go unreported. So just a quick little history lesson. Back in the 1950s, B. Series was established as the causative agent of a mild form of diarrheal illness characterized by symptoms of diarrhea and abdominal pain. But in 1971, clinicians in the UK reported a completely novel foodborne illness in people who had eaten cooked rice served at takeaway restaurants. And interestingly, microbiologists were able to link this illness to B. Series. And this was an emetic illness, and it was characterized by symptoms of vomiting, and it had a much shorter incubation time than this diarrheal B. Series illness. And later, scientists discovered that this emetic form of illness was caused by a toxin called cerulide, which is produced by an enzyme called cerulide synthetase. And the genes encoding cerulide synthetase were found to be located on a plasmid. So at some point in time, B. Series apparently gained this plasmid and gained this ability to cause this new form of emetic disease. And a couple early studies reported that all of these cerulide producing B. Series strains that are capable of producing this emetic toxin, which I'll refer to as emetic B. Series, they reported that they belonged to a highly clonal lineage. So it was hypothesized that at some point in time, this lineage gained the ability to produce this emetic toxin, and this toxin provided emetic B. Series with some sort of evolutionary advantage, which allowed it to proliferate. However, other early studies asserted that emetic B. Series was more diverse than that, and that multiple lineages have acquired the ability to produce this toxin. So this kind of got me thinking, like, what exactly is emetic B. Series? Is it the single lineage that acquired the ability to produce cerulide one time and then it proliferated, which I refer to here as the static hypothesis? Or is emetic B. Series actually just a collection of multiple lineages that acquired the ability to produce emetic toxin? Maybe lineages can even lose this ability. And I refer to that as the dynamic hypothesis. So to investigate this, I aggregated genomic data and metadata from literally every B. Series and B. Series-like genome ever sequenced. And so rather than overwhelm you with like a massive phylogeny with thousands of genomes, here's just a small phylogeny constructed using a tiny subset of the genomes, but this gives us our answer to our question. So this is a time-scaled phylogeny of B. Series genomes that belong to what's called the ST26 lineage, which is considered this high-risk lineage. And we have year plotted along the x-axis here, and then the tip label colors denote whether a strain is capable of producing emetic toxin in pink or not in green. And over the course of its evolutionary history, this ST26 lineage acquired the ability to produce emetic toxin multiple times, so at least 3 separate times within this lineage alone. And not only that, emetic B. Series can lose the ability to produce this emetic toxin, and this has happened at least 3 times, even in just this one lineage. Which means that there's strong support for this dynamic emetic B. Series hypothesis because the ability to cause emetic illness appears to be something that can be gained, it can be lost, and it can happen across multiple lineages. Additionally, we predict that some of these lineages acquired the ability to produce this emetic toxin before the emetic illness was even reported and discovered in the 1970s. So what this means is that emetic B. cereus was likely responsible for illnesses prior to the 70s when it was first detected, but these illnesses probably went undiagnosed or unreported or unattributed to B. cereus. So overall, just kind of a cute little case study of a pathogen that acquired the ability to cause a completely novel illness and how we can use publicly available genomic data and metadata to kind of get some insight into its evolutionary history. Also note that if B. cereus isn't your organism of choice or virulence factors aren't really your thing, these sorts of models that I use to carry out these types of studies can be applied to many other types of organisms, not only bacteria, but also viruses and higher eukaryotes as well. And you can model any type of discrete traits, so not just toxin production, as long as there's some sort of signal. But back to emetic B. cereus for just a moment. The whole reason I was able to carry out this study was because I knew exactly what I was looking for. I knew that if a B. cereus strain has the genes encoding cerulide synthetase, it could produce cerulide. And if it could produce cerulide, it could cause emetic illness. But this took decades to figure out. First, clinicians and epidemiologists and microbiologists had to identify the disease. Then they had to link the novel disease to the pathogen. And then it took decades for scientists to identify cerulide, the actual toxin responsible for this illness. And then finally, over a decade after that, scientists were able to identify the cluster of genes that encode cerulide synthetase, which is responsible for producing cerulide. Then I, the freeloading computational biologist, get to piggyback on all of these efforts and like decades of work and just simply search for these genes in whatever my collection of genomes may be based on sequence homology. So with this rule-based approach for virulence factor identification, I had to know exactly what I was looking for if I wanted to find it in a genome. And this is all fine, but what if I'm dealing with a truly novel emerging pathogen that's never been seen before, and I don't have decades to figure this out? Or maybe I'm dealing with some really complex disease that's the result of microbial species interacting with each other, or maybe interacting with the human host. So in these sorts of scenarios, this rule-based approach probably isn't going to cut it because I don't know what I'm looking for. And so that brings me to the second part of my talk, which involves a novel method for identifying novel microbial secondary metabolites. And so if you've never heard of them before, secondary metabolites are compounds that aren't directly involved in the growth or development or reproduction of the microbes that produce them. But these compounds can play really important roles by allowing producer organisms to adapt or respond to stressors or stimuli in their respective ecological niche. And if this concept is a little vague or confusing, don't be worried because you've definitely heard of some secondary metabolites before. So for example, cerulide, that emetic toxin that some B. cereus strains produce, that's a secondary metabolite. Another example, if you're a Salmonella fan, because I have a feeling there's a couple of those here, Salmonella's O antigen is considered a secondary metabolite that allows the pathogen to interact with the human host. And these are just a couple more examples. Many other secondary metabolites are important virulence factors or toxins. And if you want more, you can head on to the MyBig database. The link is here. Type in your favorite pathogen's name, and you'll probably find something you're familiar with. However, not all secondary metabolites are bad guys. Many important antimicrobial, anti-cancer, antifungal, anti-diabetic drugs have been derived from microbial secondary metabolites. And as I kind of alluded to earlier, at the level of the entire microbial community, secondary metabolites allow microbes to interact with each other as well as with their environment. So for example, within the context of the human microbiome, microbes in the human gut have been found to produce a wide array of secondary metabolites that allow them to interact with each other, as well as the human host. And the action of these metabolites in combination with other factors can affect things like host immune system modulation, the efficacy of drugs taken by the host, and some can even play a role in carcinogenesis. So these very small metabolites produced by microbes can actually have really big impacts on human health. And as it turns out, the enzymatic machinery responsible for producing many secondary metabolites that microbes produce are encoded by physically clustered groups of genes called biosynthetic gene clusters, or BGCs. So if you can kind of see where I'm going with this, if we can identify novel BGCs in microbial genomes, we can possibly find novel secondary metabolites. And if we want to search for BGCs in a set of microbial genomes, it's perfectly acceptable to use a rule-based approach like BLAST, or you can use HAMMER, or BGC-specific approaches that rely on these types of methods. If you've ever heard of like anti-smash or PRISM, that's kind of what those are doing. For example, these are the sorts of approaches I use when I'm looking for BGCs like cerulolide synthetase in a set of Bacillus Series genomes. So totally acceptable, valid approach. But if we want to prioritize novelty and find BGCs of unknown architecture, an alternative approach we might take would be to collect as many examples of BGCs as possible, along with genomic elements that aren't associated with secondary metabolism. And instead, we could train a model that learns different protein domain patterns associated with BGC and non-BGC regions. And once we train our model, we can use it to calculate the probability that a particular gene in a genome or a particular protein domain in a gene is part of a BGC or not. And we call this a model-based or machine learning-based approach. And this is literally, in a nutshell, what I've been doing for the past few years. Sadly, I don't have time to get into the nitty-gritty details, but my colleagues at Embl and I recently developed, and we're currently finalizing, a machine learning-based approach for novel BGC detection called Gene Cluster Prediction with Conditional Random Fields, or GECKO, which uses a type of discriminative graphical sequence learning model called a conditional random field, if you've ever heard of those, to identify BGCs in microbial genomes and metagenomes. You can check out the preprint for more details. We show that it's both more accurate and like nearly an order of magnitude faster than current state-of-the-art model-based approaches. But just a warning, it's a methods paper, so it's probably not a super fun read. But rather than show you a bunch of just like ROC curves and precision recall metrics that we came up with, I thought it'd be more fun to quickly mention some recent applications, because most people here are probably interested in infection biology. So keep in mind, this is all very new and it hasn't been subjected to peer review. Take it with a grain of salt. But we applied GECKO, our tool, to over 39,000 isolate genomes of species found in the human gut, as well as over 280,000 metagenomes derived from the human gut, to kind of gain some insight into the biosynthetic potential of the human gut microbiome. And overall, this produced like, I think, over 3 million BGCs that we've had to deal with, which is kind of a challenge. But here you can see a two-dimensional representation of these 3 million plus BGCs that we detected in the human gut microbiome. Each of these dots represents like a cluster of BGCs, and dots that tend to be closer together in space represent BGCs that are more similar in terms of their protein domain composition. And dots shown in these colors over here are experimentally validated BGCs. We already know they exist. We know they produce a secondary metabolite. And they're just kind of like superimposed on top of this gut microbiome landscape. And then dots in this gray color here are BGCs detected by GECKO, our tool. And so the main takeaway of this like ink blot looking figure is that there's a lot of BGCs that GECKO detects that are pretty distant from known experimentally validated BGCs, meaning that there's huge portions of the human gut microbiome biosynthetic landscape that we've just like never even touched. So now, in addition to fine-tuning the method, my current work involves actually narrowing down some BGCs that our experimental collaborators can test in vitro. So this is cool. Exploratory stuff like this is fun, but identifying novel virulence factors or disease associations is probably more fun. So as kind of like another exercise, we also very recently decided to look at colorectal cancer, or CRC, because the microbiome, in combination with other factors, is known to play a really important role in CRC. Also, that's what my PI studies, so I had to do it. And previous studies have shown that numerous microbial species are enriched in the microbiomes of people with CRC. But rather than just look at species enriched in CRC, why not look at BGCs enriched in CRC? Because BGCs represent functional units rather than taxonomic units like species. So super recently, like last week, we applied Gecko to a couple data sets affiliated with a recent colorectal cancer meta-analysis. And we were able to identify over like 164 BGCs that are highly enriched in the microbiomes of people with CRC, as well as 15 BGCs that are highly depleted. So they have a negative association with CRC. And here's just a plot of these different BGCs we identified and the degree of enrichment in terms of CRC associated fold change. So positive fold change means CRC associated and negative means CRC depleted. And they're colored by these different biosynthetic classes if you're familiar with those. So currently we're having some collaborators looking into these, which is super exciting. Sadly, that's all I'm allowed to tell you right now. It's just like a little sneak preview, but you can do cool disease association stuff with this. Also, if CRC isn't your thing, these methods can be easily extended to other systems. For example, if you want to look at microbe-host interactions for other diseases or other human host sites, you could hypothetically do that. Also, it's not even, you don't even need human data. We actually recently collaborated with some ocean microbiome researchers who are interested in finding novel natural products. And we applied Gecko to their ocean metagenomic data set. So that's pretty cool. So insert your favorite biome, microbe, or pathogen here. There's a lot of ways you can extend this. So that was probably a lot, but hopefully this kind of gives you just an overview of this like cool sorts of questions we can answer using publicly available genomic data and metadata alone. Everything I just showed you was in silico, and it was all performed using existing data and metadata with no additional experimental work done. So in conclusion, we can leverage the vast amounts of publicly available genomic data and metadata to gain important insights into pathogen evolution, transmission, interactions, or what have you. But it's also essential to note that sometimes or often in silico approaches alone can only get you so far, which is why collaboration is essential, experimental work is essential, and data generation, curation, storage, maintenance, those are all essential because none of this would have been possible without any of that. Just a quick special thanks to all of my collaborators, especially my PI, Georg Zeller, who's the brains behind the initial concept of Gecko, and my PhD student, Martin Lerald, who easily could have given the Gecko part of this talk probably better than me, and to all my experimental collaborators, because like without them, I'm beholden to them. And thanks to all of you for listening.
Thank you very much, Laura. This was a great reminder what the enormous value of these millions of microbial genomes is, and how we can exploit them. So fantastic prospect here. Thank you very much. We've got a few, and for you allowed some extra time now, so we can get a few specific questions here out of the way. Mariana Pires Braga asked before you turned to the gene clustering, what kind of model you used to infer the gains and losses of the emetic trait, and did it affect diversification dynamics?
Good question. Sorry, you guys can hear me, right?
Yes.
Very cool. Sorry, I forgot if I unmuted myself and I can't see the button now. So for this sort of thing, the one I reported in the paper, you could just use a parsimonious approach. It did the same thing. I used a maximum likelihood approach as well, like ancestral state reconstruction in, I think it was like Phi tools, but there's also Bayesian approaches that show the same thing. That I tested out. I don't think I reported those, but basically in the case of Emetic V series, like it was obvious that this was being a trait that was being gained and lost. So let's put it that way. And that was only just one lineage, like there were more lineages than that. So yeah, a couple different approaches and they all kind of gave the same answer, if that makes sense. Sorry, and the Bayesian approach, if you're interested, if you like Beast, it's you could do this in Mascot, or you could do this in BDMM. So like a structured coalescent or a birth death migration model framework, you could do that. And those are the models I work with these days, but I didn't report it in the paper, I don't think.
And did it affect diversification dynamics of the genomes, I suppose, that you were studying?
What is meant by that? Like, oh yeah, okay, so sorry, I did think about that for a second. So not sure. I guess I didn't really, I didn't look at that, but that's actually a really good question.
So while we're talking about that toxin, I wondered whether you can tell where it was gained from, and whether you can predict that, and then even perhaps preempt it in some way, use that knowledge.
That's a great question, because, and my apologies if there's like an Emetic B serious person on the call and I may have missed your paper. I don't think they really know exactly where this toxin came from. It is a soil dweller, so I think people hypothesize that it's something to do there. The idea behind Gecko would be to hopefully like identify stuff like this, maybe in like genomes that people are sequencing that haven't come into, I guess, haven't been like found in humans yet. And then hypothetically, I guess you could try to predict if something could be a threat. But like, which I think that's a cool idea, but I haven't gotten that far into it. But good point. And
one question from Teresa Friesan, now concerning the BGC regions. How did you select for the non-BGC region? Through position relative to BGC or some other way?
Oh gosh, that's a great question, because that's actually really difficult and non-trivial. So basically the way we and other people do it is we use some sort of method to identify known BGCs in whatever genomes you're interested in using for your training data set, and then you mask those. Other people use BLAST, we used anti-smash, but exactly at the end of the day, it's like you're kind of dealing with a situation where you're working with negative data, and it's like you can't 100% be sure that it's like they're true negatives. Like you could find some secondary metabolite that you're calling like, or some BGC that you're calling like a non-BGC region when you're training. So then obviously like that wouldn't be ideal, you'd miss that. But basically you do it using relative to known BGCs. You have to mask them and then construct your training data set that way.
And one last quick question, Niklas Jarboy asks whether you are, what you think about the quality of the available metadata that you find with bacterial genomes.
That's like a great question. So I guess it depends. Like ideally you have like, you know, studies that you can link it to a publication and a paper. For the Bacillus Series stuff, I literally like with that small lineage, because there weren't that many genomes in this one particular lineage, I went through every single genome and looked up a paper and tried to make sure I was like extra careful. But at the end of the day, it takes a lot of curation and there can always be mistakes. And like, so for the Gecko stuff, I'm kind of like, you know, beholden to, I guess, what people label their metagenome, if it's CRC or not CRC associated. So yeah, there's little steps you can take to hopefully link it back to papers, but yeah, at the end of the day, you know, more genomes, more problems, but also more insights.
Well, thank you very much, Laura. We need to move on. So while you unshare, I will introduce our next speaker, but just remind everybody that there are, of course, many bigger questions we can now discuss about how we can use these technologies Laura has just talked about to get at the biology of completely unknown and intractable organisms that I found very interesting, and perhaps many other bigger questions that you want to ask. And do put them into the chat as well, so we can save them for the end of this session. Our next speaker is Professor Jay Hinton, who is Professor of Microbial Pathogenesis at the Institute of Infection, Veterinary and Ecological Sciences at the University of Liverpool. And Jay's lab uses transcriptomics to understand the virulence of new variants of Salmonella in sub-Saharan Africa, and also to investigate pathogenesis and infection biology of these bacteria. So please, Jay.
Great. Let me just get the sharing working and then the slide. Good. Well, lovely to be here. I have many happy memories of spring visits to Umeå, Uppsala and Stockholm, and I'm really sorry not to be there with you, but perhaps next time. Yeah, well, you're right, Oliver. That's exactly what I'm going to be talking about. But I thought I'd begin with just the opportunity of showing you a snake. This is a puff adder from a collection at the Liverpool School of Tropical Medicine, where they're kept for their venom. It turns out all the Salmonella, all the snakes in Liverpool, which came, the venomous snakes in Liverpool came with their own Salmonella that they picked up in Africa and brought back for us. So we looked at 240 snakes and we found about 100 different types of Salmonella. But that's another day. And I'm going to move on to what you all think about when we think about Salmonella, which is gastroenteritis. And the type of Salmonella that causes this gastroenteritis is called NTS, non-typhoidal Salmonella, and it really occurs around the world. Major culprits are Typhimurium type ST19 and also Enteritidis, which I won't be talking about today. Another famous type of Salmonella causes typhoid. This is really in the developing world associated with poverty, without good sanitation, without good drinking water. So we can understand which parts of the world this is happening with. Killing around 200,000 people a year. And the main types of Salmonella are called typhi and paratyphi. But it turns out there's a new type of salmonellosis called invasive non-typhoidal salmonellosis, or INTS. This is an emerging disease and it's rather lethal. It's a multi-drug resistant pathogen, so its antibiotic resistance causes big problems, and the case fatality rate is as high as 20%. So it's a big concern, particularly in sub-Saharan Africa. These are the numbers. This is the mortality associated with this disease in sub-Saharan Africa, and the numbers are about 500,000 people were killed over the past decade by this disease, and about 2/3 of them by Salmonella typhimurium, which is what I'll be focusing on today. The people it targets are people who are immunocompromised, people with HIV or malaria, or people who are malnourished or have anemia, of which unfortunately there's quite a lot in sub-Saharan Africa. Some fantastic work from the Sanger Institute, from Gordon Dugan's group about 13 years ago, looked at the genomes of a few isolates and proved that they were not the ST19 that caused gastroenteritis. They were a completely new type of typhimurium called ST313, which is what I'll be focusing on. So this is what we understand about the population structure of Salmonella typhimurium. We have many which are generalists. They can infect animals and people. Some orphans that get into birds and can't come out again, so they specialize to birds. ST19 is one of these generalists associated with food. And this new sequence type associated with bloodstream infections only found in Africa. And when I saw this, I realized that this pathogen must have evolved in an interesting way, which I needed to understand. Now, we're very lucky with Salmonella because people have been studying its virulence for 50 years, both of Typhimurium ST19 and typhi. We know about the SPY1 pathogenicity island, the SPY2 pathogenicity island, how some of the effector proteins produced by these interact with all sorts of parts of this cellular machinery. But that took 50 years. How are we going to do this for a new pathovariant of Salmonella, this new ST313? As Laura said, we don't have 50 years. And we know a lot about the hosts that Typhimurium likes to live in, be it cows or chickens or pigs or mice or even snakes. But actually this pathogen seems to have adapted to live in humans. So lots of what we knew in the past wasn't really relevant. Now we're very lucky. Salmonella turns out to be the most sequenced bacterial pathogen. If we look at what's happened since 2017, we can see there's a huge explosion in genome sequencing. We're now up to 359,000 genomes, all available in this database Mark Axman developed called Enterobase, where you can both look at the genomes and you can do phylogenetics and make trees. It's a fantastic resource. So this is the backstory. And Mark's group have used this. They've classified it into different bubbles of different types of Salmonella. The major ones are Enteritidis and Typhimurium because they cause most human disease, so they've been sequenced most. We've also got paratyphi and typhi, etc. Okay. So how are we going to do this? And as Laura said, genomics is hard, and you can't do everything with genomics. This is a graphic put together by the Wellcome Trust 25 years ago, explaining how you take a genome, and use bioinformatics as the glue to stick together the transcriptome, the proteome, and a mass mutagenesis approach. And I can only have time to talk about some of this today. The first Salmonella ST19 genome came out in 2001, and so that's really when this sort of fun started. We can begin with a pairwise comparison where we look at the ST19 genome we know a lot about, which has a major virulence plasmid, and then we can compare it with the African strains we're interested in, coming from the lineage 2 of ST313, a strain called D23580. The same virulence plasmid, but with an antibiotic resistance cassette encoding several antibiotic resistance genes and some novel African plasmids. Overall, the chromosomes are 96% identical, so they're really quite close. There are about 300 gene differences, largely due to plasmids and prophages, and about 856 nucleotide differences. So if you look at the circular chromosome with Salmonella, and we're asking about, we're interested in things like the prophages, BTP1 is my favorite. We can look at pathogenicity islands like Spy2, and then we can ask where all the SNPs lie when we compare the African strain with the European strain. And I was wondering whether they might all be clustered on one part of the chromosome, perhaps around some particular virulence factors. Well, they're not. All the SNPs occur all the way around the chromosome. And when we look at these 856 SNPs and we look for which are the indels, which cause stop, which cause missense mutations, etc., there's really no clue to function just by looking at these. So at that point, we wave our hands and try to wonder, well, which one of these SNPs is important? We can't tackle them all experimentally. So transcription is transcriptomics is the way we have approached this, where we can look at all of the Salmonella transcripts in one experiment, either during in vitro growth or inside mammalian cells using a technique that I developed when I was collaborating with Michelle Carroll at the Karolinska. We published this, gosh, almost 20 years ago, and it gives you an opportunity to get a Salmonella transcriptome from inside a macrophage, which is a real infection, which is fantastic. And this concept of functional transcriptomics, I thought I'd mention briefly. I'm really only going to be talking about Rna-Seq today, but the things we are interested in is how a transcript is regulated. So we do differential Rna-Seq to identify transcriptional start sites. And we also ask questions about biological relevance by using infection models and TN-seq or TRADAS. Okay. So the sort of gene expression approaches we were using were really modeled on what the pathogen is experiencing in the mammalian host. We know there are rapid changes in pH when the bacteria hit the stomach. And we know that to survive, the bacteria have to be fit, which means that most of the virulence genes that are required, say, to infect the gut are not expressed when the Salmonella is sitting on a lettuce leaf. And to optimize fitness, Salmonella has to express the right genes in the right place at the right time. So I was determined to understand that. So we came up with 21 infection-relevant conditions involving acid pH and bile, changing of osmolarity, taking away oxygen as you do in the gut, and also related to life in the macrophage, because Salmonella can live inside human macrophages within a vacuole, where they're exposed to a different acidic pH, reactive oxygen species, reactive nitrogen species, a low magnesium environment. And to do these without perturbing the growth of the bacteria too much, we use 10-minute environmental shocks. And this approach now being used by collaborators of ours and many different pathogens, I think is a great way to see how bacteria start to adapt to particular environments. So we first did this for ST19, a strain called 474. We looked at all of the different conditions. Here are our 20 conditions. Here is the percentage of genes which are expressed. And overall, about 86% of the genes were expressed in one or more of these different environmental conditions. This has all been published. It's all made so you can Google it. If you go to SalCom-Mac, Salmonella Compendium Macrophage, it's designed to work on a smartphone in a pub, and it will generate heat maps on the fly for any genes you're interested in or all of the virulence regions of Salmonella. Okay. So our hypothesis about this African variant was that Some of these genetic differences would change the transcription of the key genes, leading to alterations of phenotypic properties, leading to the severe disease outcome. And based on what we knew about typhimurium and particularly typhi, we expected the pathogen to be trying to evade the immune system using some type of a stealth phenotype. Because what Salmonella usually does is it goes in and it is taken up by macrophages or dendritic cells, and it's by living in them or perhaps avoiding neutrophils that is able to cause disease. So a bit of RNA-seq data. I'm showing you the RNA-seq data for the positive strand, the genes going from left to right, and the negative strand underneath, the genes going from right to left. Lots of color means lots of transcripts mapping to particular genes. Here we see some being expressed. Here we see some other genes being expressed in the minor strand. And if we compare the European strain with the African strain, we get a very similar picture. You see these green blobs here, seen in the African strain. We see these green blobs here in the European strain, seen in the African strain. So there's many similarities at the transcriptomic level between the African and the European variant. In fact, 99% of the transcriptional start sites were conserved. So of course, we had to build on what was done in ST19 and do a lot of environmental conditions using our African variant. This work was done by Rocío Canals using the same conditions we used for ST19. We ended up with a new website where you can go and look at both the European and the African variant in parallel and make your heat maps on the fly. This time you Google for Salcom D23580. And thankfully, there's no other one of those in Google. Okay. After doing quite a bit of RNA-seq, we did more replicates to really improve the quality of the data. We focused on just 5 conditions with our replicates, including inside a macrophage. And we found about 670 coding genes in small RNAs that were differentially expressed. The red genes are upregulated in the African variant compared with the European variant, and the blue genes are downregulated in the African variant. But which ones are we going to focus on? There is a lot of noise in that data. And what we decided to do was look for genes which are upregulated in every condition. We found 6 genes. Then we wanted to find genes that are upregulated and also have mutations, SNPs in the promoter region, because I always hoped that we'd find a mutation in a promoter region. And the one that we came out with as being most interesting was Pgte. Pgte is something you won't have come across before. And this is where Disa Hameloff comes into the story. Disa was a Werner Gren Foundation funded postdoc who did some fantastic work helping get my Liverpool lab set up and getting the RNA-seq working in this African variant. Okay, in the European variant, there's not very much transcript. You don't see very much color for the Pgte gene. In the African variant, you have lots of color, meaning lots of transcript. So we knew the Pgte gene was being highly expressed in the African strain. What is Pgte? Well, it's not been worked on that much. I had to commission an artist to make this. This is the Pgte omptin protease sitting on the outside surface of the Salmonella. Here's some short LPS, outer membrane and the inner membrane. And the active site of the Pgte is here, and it allows the protein to cleave, the protease to cleave all sorts of different proteins. I don't always believe transcriptomics. Of course, we had to do proteomics. We did a comparative analysis of the same two strains. We found 1,800 proteins. We asked which ones were differentially expressed. All of these red proteins, we're going across the 4.5 megabase Salmonella chromosome here. All of these red proteins are upregulated in the African strain, and the blue proteins are downregulated in the African strain compared with the European strain. And what is the most highly upregulated protein is Pgte. So this was not just happening at the transcriptomic level, it was also happening at the protein level. So we had to think about the promoter. We know a lot about promoters in Typhimurium. We've defined them previously. We know exactly what the major tartar box looks like. And we found for Pgte in the African strain, there was a T nucleotide here at -10, and in the European strain, there was a C nucleotide. So for those of us who think about T nucleotides encouraging optimal binding of RNA polymerase and high levels of transcription, all we needed to do was to put the C nucleotide into the African strain at this location. So it's a bit of single nucleotide engineering, and it's much, much easier to do this in PowerPoint than it is in reality, and Disa will attest to that. We call this SNP the Pgte European SNP. So the first thing DESA did was to use a qRT-PCR approach to compare the Pgte transcript in the normal European strain and the normal African strain, so much more expression in the African strain. And then making this one nucleotide change, suddenly you're downregulating the expression of the Pgte gene, fitting with what we thought. This was also repeated at the protein level. So we need now to do some real experiments, and really the data we have used is to excite us about doing particular types of experiments. The one we decided to focus on was being killed by human serum, and African Salmonella is able to resist being killed by human serum associated with it being a bloodstream pathogen. But when we change a single nucleotide, we suddenly make the strain extremely sensitive to being killed by human serum. And we know this is associated with a Pgte gene, because when we delete the Pgte gene, we get the same serum sensitive phenotype. And what is it in serum that's killing the Salmonella? Well, if we heat treat the serum, which destroys complement, suddenly there is no serum killing. So we know that this killing was being mediated by complement. So at that point, I had to rush to remind myself what the complement cascade was, these interesting stages of the process, C3b deposition, ending up in formation of a membrane attack complex, which will break the bacteria open. From work that's been done previously, we knew that Pgte would interfere with the alternative complement pathway because it can block formation of the C3b complex, which meant our next experiment was fairly obvious, a factor B cleavage assay using our African Salmonella. So here's the wild-type African Salmonella cleaving the 90 kilodalton complement factor B and getting this 57 kilodalton cleavage product. We change one nucleotide and there's hardly any cleavage, and we delete Pgte and there's no cleavage. So the single nucleotide is responsible for high expression of Pgte and hence cleavage of complement factor B. So now we can look at our huge amount of SNPs on the chromosome. We've actually found the one that's causing a significant virulence phenotype, this C to T transition. And this is the take-home message from this Pgte story. With a C nucleotide, there's little transcript, there's little protein. You have bacteria in the human bloodstream which are burst by the attack of complement. Whereas the African Salmonella with a T nucleotide, lots of transcription, lots of protein, and here's a happy Salmonella with loads of Pgte on the surface moving around in the bloodstream. Okay. Now that was what we could do when DESA was in the lab. We used genomes that had been mostly isolated in the early 2000s. There were only 129, and they came from 7 countries, mostly in Malawi, and we were concerned about sampling bias. So we needed to take this to the next level. What relevance is this for African patients today? We need to bring ourselves up to date. And does the presence or absence of this SNP, this nucleotide change, represent an evolutionary landmark? So because we wanted to move beyond these number of isolates, kicking and screaming, I set up a 10,000 genomes project so that we could do Salmonella bloodstream isolates from around the world. And Blanca Perez Sepulveda was the postdoc for my group who led this work brilliantly. And Neil Hall at the Earlham Institute, an old friend of mine, also came up with a protocol which allowed us to sequence bacterial genomes for €9 each, including DNA extraction. Remember though, this was done with 10,000 genomes, so obviously we're working at scale. This has just been published in Genome Biology if you're interested, and I haven't got time to go into this in detail, but we only sequenced genomes from isolates for which we had good metadata and for which we had already had some AMR sensitivity information, and they go really back for the last 2 decades, but the first is from 1949, so we can get some historical perspective. Now, putting together a lot of isolates from Malawi, from a research center involving the University of Liverpool and the Wellcome Trust, and also Francois Xavier Weil's collection from the Pasteur Institute, going back to 1949, we ended up with a picture looking like this, where we started with samples coming from West Africa, and then over time, we were sampling more countries, until in the end, we went from the Mediterranean all the way down to South Africa. If we look at what the isolates were, the metadata was appalling at the beginning, and then as things improved, we had a lot of stool isolates, and then this epidemic of bloodstream infection started in the late 80s at the same time as the HIV epidemic, and we had many more bloodstream isolates. And if we ask questions about what is it about these isolates, well, antimicrobial resistance arose in about 1987, first of all ampicillin, then chloramphenicol, and then finally cotrimoxazole, making these difficult to treat clinically. So I've told you about this nucleotide change. I've told you that it was important in a couple of strains. What happens when we look across all of the isolates from Malawi between 1996 and 2016? And remember, we're looking at this T nucleotide from the African strain, or the C nucleotide, the blue one, from the European strain. So the C nucleotide was found at the beginning of the 2000s and then disappeared. This is the percentage of isolates in Malawi, and it was replaced by the African SNP. This T nucleotide was found in all of the isolates we looked at, which I was delighted by and slightly surprised by as well, which is very good news. We also use this to do something I know Oliver wants us to talk about, which is prospective sequencing for surveillance, and discovered that the lineages have been changing in Malawi. The one we have been working on has been reducing. There's a new one that will be publishing soon, and a lineage 3, which is antibiotic sensitive, which we wouldn't have found otherwise. So we think the Pgte mutation increases the serum resistance of these bacteria. We think the bloodstream infection was the evolutionary driver, and it's the combination of this SNP with another pseudogene and multi-drug resistance gave these bacteria the competitive advantage right across sub-Saharan Africa. So to summarize, you can do a lot with genomes, both looking at relatedness and virulence, but by doing functional transcriptomics, you'll find non-coding genetic changes in promoter regions that can be functionally very important. Just a last advert, we've just got a new grant from the Wellcome Trust to take the African Salmonella project to the next stage. I'm looking for 2 molecular micro postdocs, an infection biologist who's used to working with human macrophages, and a computational biologist, and an international student to come and do a PhD. So please let people know. They should email me. I couldn't do this without our collaborators in Malawi, and particularly Ben Kumwenda, who's really helped a lot with the genomics in recent years, and the clinical team, and an amazing group I've had working in Liverpool. Disa was very important. Carsten, Nico Wenner, the best geneticists I've worked with. Rathia Canals, who got the Rna-Seq working. This is the group and the funders, including the SNSF and the Venegren. So I'm very grateful for your time, and I'd be happy to answer questions now or in the breakout.
Thank you very much, Jay. This is fantastic. This really adds a new perspective of how you gain traction by having large numbers of the same species, basically, of different strains, the same species, to get to pathogenicity, which complements what we've heard before. Very nice. And I made a special note, of course, of what the benchmark is for a piece of software or a database. It has to work on a smartphone in the pub. So thank you very much for clarifying that.
Yeah.
Because that really gives us a good argument to work on the pub culture in Sweden, right? and see how much funding we can mobilize for that. So fantastic. I completely agree. Let's see. We've got one quick question in the chat here. Let me quickly navigate there. I've just lost it. Where
I've got it. I can see a question about infecting rodents. Yeah, people have done experiments in susceptible mice, looking for a different phenotype to your normal gastroenteritis associated salmonella. Generally, the response is very, very similar, but we see more inflammasome induction by the gastroenteritis associated salmonella than the African salmonella. So we think this is indicating a stealth phenotype where the pathogen can hide from the immune system. The other thing that was seen in mice by Denise Monach's lab is that the African Salmonella can be disseminated in dendritic cells in an effective way due to the pseudogenization, making a pseudogene in a single effector protein called SSEI. So if you knock out SSEI, suddenly it makes African Salmonella able to be disseminated effectively and cause systemic infection in the mouse. But it's a really difficult model to use. And we decided we really couldn't do effective experiments that would differentiate the African and the European variant.
Great, thanks a lot. Let's turn to our last speaker before we have many, because we return to the general questions that were raised here so far. I'm super happy to welcome Jeff Barrett to Sweden. He's on the one hand the CSO of Nightingale Health, but he's previously been the director of the COVID-19 Genomics Initiative at the Wellcome Sanger Institute. And among the many things that Jeff does seemingly without effort is to move between the academic and commercial sector, as you see just there. And I'm not surprised by that. I know Jeff as a human geneticist who gets really excited about statistical problems and who is a strong advocate of open science, but who also always wanted to generate actionable outputs and that benefit human health. So I bet this will have influenced his response to the COVID-19 pandemic. But I better let him tell you about this himself. And he is going to talk about tracking the rise and fall of SARS-CoV-2 variants in the UK with over 2 million viral genomes. Jeff.
Thanks very much, Oliver. Yes, I'm going to talk about work I did over about 18 months or a bit more, where I kind of came back to the Sanger Institute, where I had previously led a research group in human genetics to work on this very large genomic surveillance of SARS-CoV-2 program that had been set up. And I'll get to this in a second, but it was certainly early on in 2020 based hugely on the effort of volunteers from across the Institute, indeed a much wider network of collaborators called COG UK, which was the consortium that was put together, led by Sharon Peacock, to deliver genomic surveillance of the coronavirus. And in particular, this is the picture as of this morning. I have never managed to give this talk twice without having to update all of the slides about how many different variants and peaks there have been of the UK's pandemic. I start the clock here at September of 2020. Before that time, the testing infrastructure in the UK and elsewhere was not sort of able to detect a reasonably large fraction of cases. So this is kind of the window in which I think this is a pretty accurate view of the number of cases per day in the UK. Of course, that's probably going to change, or indeed is already changing again, as the UK, like many other countries, is reducing kind of universal access to free PCR testing, which means that again we'll be in a world where we don't have complete ascertainment of ongoing cases. But of course, and really nicely sort of transition from the end of the previous talk, that underneath this is not just one monolithic virus that is coming and going. It's actually been a very diverse set of different waves of different variants of this virus with different biological properties. And this surveillance effort really brought together, and it was put together in the middle of 2020, a number of key pieces. So for example, we at Sanger, so as I mentioned, we were part of a sort of UK-wide initiative, which involved, for example, many sequencing sites working with local hospitals. What we did at Sanger, which is a big genome sequencing institute near Cambridge in the UK, is to set up a collaboration with the centralized PCR testing around the UK. So there were originally, I think, 3 sites, and it grew to, I think, 7 or 8 or something over time. But basically, it enabled us to do something because anybody who'd sort of went to a car park testing site or got a home testing kit and sent it back, the PCRs were done in a small number of centralized places, and that meant we could essentially set up these vans every day to be driving up and down the country to just a small number of places and bring the waste material from the PCR tests to the Sanger Institute to be put onto a very high throughput sequencing pipeline. So we had to kind of stick a big freezer in the car park that we could be unloading these boxes and boxes of sample. They, as I said, literally what was inside of these was directly the kind of, sorry, my slide's not advancing. Oops, there we go. Directly the the RNA after the PCR test. Initially, we were doing the cherry picking of positives on site, and eventually that was moved back into the testing lab. So Sanger is just getting huge volumes of positive tests. We had to build a number of different very large pipelines to make this happen, to both process the samples, to prepare the libraries for sequencing. We used a version of the Arctic amplicon-based sequencing protocol, which was developed by other colleagues in COG UK and kind of optimized for this high throughput scenario. So basically that corresponds to taking about 100, approximately 400 base overlapping chunks that have primer pairs. And so you do a big multiplex PCR reaction to amplify the whole genome so that then you can get the complete sequence in these overlapping chunks. And there are some sort of tricky bits with that, but generally speaking, it actually was possible to make this work at very, very large numbers. And we pooled them and sequenced them on Sanger's high throughput Illumina pipeline. And the numbers, in 2020, we started to kind of get the hang of this and do some hundreds or thousands per week. But actually at the height of production in the beginning of this year, the Sanger was producing something like 60 or 70,000 complete genomes per week, week after week. So it generated to date over 2 million coronavirus genomes from that kind of range of the pandemic, from about the late summer of 2020 until now. Again, there's even another set of UK genomes that came from other providers on top of that. And certainly when you put all that together, it's by far the biggest single country's contribution to public coronavirus genomes. These ones at Sanger, which I should have noted in passing, they are chosen, and we got sort of better and better at this, to be as random as possible, representing the circulating community variants of the virus. And every week we update this website, covid19.sanger.ac.uk, with essentially the frequency of different variants of the virus in different local areas of England. And so you can go and kind of browse around and change the date and see when variants were coming and going. I will note as well that Richard Goder, the software developer who built this, was very focused on making it work pretty well on a mobile phone as well. So you should be able to play with this in the pub on your phone also. So really, in one sense, what this allowed us to do, I mean, there's a ton of this data gets disseminated, and there's lots of people thinking about how can you do, more classic phylogenetics and phylogenamics with numbers of sequence of this size. And there's a lot of other detailed use cases you can think about exploring specific outbreaks, for example. But one thing which I'm not sure if it was expected at the beginning of the pandemic, but which has clearly turned out to be true, certainly in the UK, is that we have been hit by repeated waves of variants of the virus with a set of mutations that change its biological properties, specifically related to transmissibility, reinfection, immune evasion, to a certain extent with virulence, and that these happen in a kind of replacement pattern. So we can see, instead of that kind of general just graph, if we take all the sequencing we've done over this period and use it to essentially infer which cases were happening from which variants at different times, we can see that the actual picture looks something like this. And I'll tell you in a little bit more detail what these colors really mean. So, so sorry, if we first look here, going up until, this is 2020, so September 2020 until November, this green color is the kind of various flavors of the virus that were very close to the original virus that started spreading from China in late 2019. And not pictured here, but the kind of in the UK, the lockdown in the spring had brought it mostly under control. We had a pretty quiet summer. There was a peak in the autumn after sort of summer holidays ended. And there was a so-called light lockdown that kept it under control. So what I'm going to show you is a series of these pictures where the top map of England is the state of the restrictions. So in November, there was this, it wasn't a kind of super strict lockdown, but there was some restrictions. And then I'm going to show you two maps that are R numbers, the reproductive number of the virus at that moment in time, which blue means shrinking and red means growing. So blue is R less than 1, red is R greater than 1. And what you can see is that these are two different lineages. So the ones I was just telling you about, those ones, the sort of original coronavirus, this state of affairs was causing case numbers to shrink everywhere in England. But this other variant, which turned out to be, now we know as B117 or the Alpha variant, was actually growing at the same time, despite those restrictions, because it had a fundamentally probably 50 to 100% higher R0 inherent reproduction number. Then as it became clear that there were some issues, there was the British government wanted to lift restrictions in December running up to Christmas. And so these green bits are sort of now regionally, they had this complicated tier system in England, in different places at different levels of restrictions. And you can kind of see that what was happening is, even though as they lifted restrictions, inevitably, even the older variants of the virus started to grow again. But certainly now the Alpha variant was growing very strongly, and especially strongly in those places that had the lowest restrictions. You can kind of see a mapping between where there were tighter restrictions and where at least there was some greater level of control. But then this led to a kind of complicated dance over the month of December, where more and more complicated levels of restrictions were introduced, and they had varying degrees of success. But fundamentally, it was only the strictest so-called tier 4, which was very close again to the complete lockdowns of spring 2020, that could really bring the Alpha variant to have case numbers starting to decrease. And so this led, the sort of delay on this led to a very large spike of cases due to the Alpha variant in the UK in sort of late December and early January. And sadly, because this was before the deployment of vaccines, it also led to a very large number of hospitalizations and deaths. And indeed, the Alpha variant then spread quite far to many other countries in Europe and indeed around the world, though it didn't quite saturate everywhere, because at this moment of evolution, there were also a few other variants, the so-called Beta variant that was first seen in South Africa, Gamma that was first seen in Brazil and spread in the US. And these each seem to have some amount of competition in different places, though Alpha ended up being the one I think that became most widespread at this point. So we saw this next peak, and then that was around the same time that the vaccine deployment really started in earnest in the UK and in other Western countries. And a combination of the lockdown and then the vaccinations meant that the Alpha wave was almost completely squished in the UK. But unfortunately, at this moment, and in many subsequent moments, this pandemic was truly global, and what was happening in other places rapidly became relevant to what was happening anywhere you were looking. And as I just mentioned, the Alpha variant that started in the UK did spread to many other places. And at this point in the spring of 2021, India was facing a truly horrendous wave of COVID. It wasn't very clear at all why suddenly, in a place where previously there hadn't been particularly bad waves, there was one that brought a very, very high burden of disease and death. And it was quite a frantic time trying to figure out whether there was a new variant that was causing this. And I remember this is a screen cap from a YouTube live stream that, at that point, globally, we didn't have, a kind of well-oiled, rapid way of releasing data for people to track different variants. And so I was watching this and froze the screen to try to count up in different states in India. This one over here, B1617, this is B117 or Alpha, which was still quite common at this moment, but you could see that this one was growing. And this is what subsequently, or a sublineage of this, subsequently became known as the Delta variant, which created an enormous wave of cases, a sort of prolonged wave of cases that had a sort of sharp peak in the summer and really just kept rumbling on all through the summer and autumn. Now, the British government put in and withdrew a number of restrictions during this phase as well. The reason being that although there were a very high number of cases, the hospitalization and death rate, while still, sadly, many people got very ill, was much, much lower than in previous waves, because by this point, the UK had vaccinated with 2 doses nearly all of certainly the highest risk population of oldest individuals, and indeed a very, very large fraction of the entire population. And Delta, even more than these previous variants, which had a combination of, although I remember at the time sort of not being able to believe it, even higher transmissibility than Alpha due to another set of mutations that partially overlapped with those that differentiated Alpha from the original coronavirus, but also was slightly more virulent. It led to, in unvaccinated individuals, higher levels of severe disease than previous variants, which obviously caused additional problems. And this led to pretty horrible waves in many parts of the world, including South Africa and the United States, where there was less vaccine coverage than, say, in Europe. And I really did think at that point that this story of having built this system and doing epidemiology on these variants might be over, but there was, with each time we had learned something about this and can see it getting a bit quicker. And this time, as people will know, the Omicron variant, which in contrast to that situation in India, where the local sequencing data was not released very quickly and it was not clear what was going on, some truly amazing genome scientists in South Africa, led by Tulio Oliveira, announced to the world that there was a new variant, this Omicron variant, which had a astonishing number of mutations, especially in the spike protein of the virus, and was spreading very rapidly in South Africa. So much that now the UK's surveillance system, we were sequencing tens of thousands of sequences, and one of a great collaborator, Theo, who was sort of watching the sequences come in one evening when we knew Omicron was coming, and this was literally the very first one in a Slack message that announced it arrived, with even just for confirmation, he pasted the FASTA right into Slack. And so here there was sort of the machine was turning quickly to be able to do a number of other kinds of analyses that are really only enabled by sequencing and having the variant type linked in close to real time. We were only sort of like 5 or 6 days behind test positive, with the sequences being in the hands of the UK Health Security Agency, the Public Health Agency. And it meant you could start to do things because we had so much sequence data that we hadn't even thought of when we originally were sort of building our surveillance and tracking mechanism, like systematically ask questions about the efficacy of vaccines between, at this point, Delta and Omicron, when Delta was going away and Omicron was arriving in the UK. And this is from one paper that the UK Health Security Agency published this year, looking at, they do it in different flavors. This is the most common vaccine regimen in the UK. So 2 doses of AstraZeneca with boosted by either Pfizer or Moderna. This is the vaccine efficacy for symptomatic disease. And you can see this is after dose 2 over time. So this is people who have different lengths of time passing since they've had their second dose. And this is after you get a booster, again, over time. You can see that we all kind of already knew that with Delta, the 2-dose efficacy was waning over time from, very high to 50% or something, jumps way back up with a boost, maybe does wane again, probably does, but not enough follow-up. But there was a real problem, which is that for Omicron, its fundamental difference, there's a debate about its exact relative transmissibility in an imaginary, totally immunonaive population, but its clear difference from the previous variants was that it was much more capable of evading immunity, either from vaccine or previous infection, to the point where people who had been boosted, who had been dosed second dose a few months ago, had essentially no protection from infection with Omicron. And even after boosting, that protection against infection did go up, but it waned pretty quickly and again became quite poor. And I can attest, as someone who had 3 doses, and even I think I was in this window of 2 to 4 weeks afterwards, me and my entire family did have Omicron about a month ago. Of course, as I mentioned, the reason that this happened, this is a kind of nice analysis where they try to create a sort of antigenic map where you project antigenic differences of different variants of the virus and different sera from people who have been vaccinated. So the big circles are virus, and the squares are individuals, sera from individuals who have been vaccinated in different ways. And what you can see is that the beta variant, which we kind of knew, and Delta variant to a certain extent, were a bit distinct from these earlier variants of the virus, both in terms of the virus itself and in terms of where people were raising antibodies after getting vaccinated. And Omicron, they need to sort of expand the scale of the graph and land it right over here, because it had, I forget, it's like 30 mutations in the spike protein alone or something. So it clearly was the first massive example of an immune jump, which led to that reduction in vaccine efficacy against infection. Now, the very good news is that the vaccine efficacy, especially in the context of boosting, is still very high against severe disease and death. So that was like the incredibly lucky break in some sense, because if that hadn't been true, I think the last 6 months would have been particularly grim. Similar paper on this riff of what kind of questions can you ask when you know such a large fraction of cases, whether they have Omicron or Delta or whatever the variant du jour may be. There's another analysis led by Tommy Nyberg. I'm not going to pronounce that correctly, given the audience is all Swedish, and colleagues from Cambridge and UKHSA. This is looking across ages at the difference in the risk of hospital admission, given that you are infected. So if people who test positive, do they go on to get so sick they need to go to the hospital? And this is the hazard ratio of Omicron versus Delta. If you the light green dots are the most important ones. If you condition on a bunch of other covariates that cause sort of spurious associations, which turns out to be very important to get a clean interpretation. So there are things like when and where and vaccine status and socioeconomic status. All of those things you can only do because you have, tens of thousands of individuals in this data set who either have Omicron or Delta confirmed. You can see that it's a substantial reduction in hospital risk for most older age groups. It's less so, and indeed not significantly different from zero for younger individuals. This is still a slightly open question. The best guess is that there's some good lab evidence that Omicron has a slightly different cell tropism, so it does more infection of the upper respiratory tract than lower, possibly causing more bronchiolitis-like disease, especially in very, very young people, say less than 2 or 3 years old, which even though it doesn't become very sick, they usually get over it, but it makes them much more likely to be admitted to hospital. So that might explain the different association in kids. But the other thing that was important about this was a sort of sub-analysis was able to show that there was a lot of press discussion about, oh, Omicron is very mild. And a part of this is true, that it does appear to be intrinsically less mild than Delta, but that was intrinsically a bit more severe than Alpha, which was intrinsically a bit more severe than the original coronavirus. So really, sort of like the common cold, it's much more like the original coronavirus, which, remember, in an immunonaive population was very, very bad. And what we could these colleagues could show was that indeed the bulk of the mildness observation in both South Africa and the UK and in many other places in Europe was about previous immunity. So lots of people who were either vaccinated or had previously been infected were either breakthrough infections or reinfections. So that creates a very large number of cases, but those were all very, very mild. But basically, it was still a very risky virus for people who had no pre-existing immunity. And as now we get to the point where even the sort of zero COVID holdouts are needing to move into a world where COVID is circulating, that difference makes a very important difference in terms of how they'll deal with Omicron. So Hong Kong and New Zealand have been examples that have recently been used, where the blue curves here are both of them essentially having to confront large numbers of cases of Omicron sweeping through their countries. But then the below the line red curves are the population normalized deaths. And you can see that Hong Kong, as has been widely reported, is having a very bad wave at the moment with a very high level of death, whereas New Zealand has had almost none. And the reason for that is almost entirely about the vaccine rate in the oldest, highest risk population. So in Hong Kong, almost 2/3 of over 80s are not vaccinated. So they were very vulnerable to this wave, whereas in New Zealand, like in many Western countries, almost all of that high-risk age group was vaccinated. And so far, at least, they are managing to kind of ride through this exit wave without a very, very high burden of death. Okay, so I'll just finish by saying we can see the UK pandemic as the sort of original coronavirus, then vanishing in front of the Alpha wave, which was very serious, but vaccines and some non-pharmaceutical interventions brought it under control to a very quiet point in the early summer of last year, which was then disrupted by the sort of rumbling Delta wave in the UK that it could never quite bring under control, which was finally pushed down by the arrival of Omicron, which led to total case numbers that I think nobody could sort of imagine happening. And I haven't even talked about this blue thing over here, which for those of you who follow very closely, there's actually 2 subvariants of Omicron, BA1 and BA2. And BA1 is apparently less transmissible than BA2. So now the UK is contending, as well as having essentially no restrictions anymore, the rise of BA2. So there will be still this graph will go on for some amount of time, and we will have to see when hopefully it will calm down to a greater level. And finally, let me just say that I played a small role in a huge collaborative team from around the UK, which was a real privilege, and it was only by bringing all those people together that we could build something like this. Thanks very much.
Thank you very much, Jeff. This was really cool to see that. I visited Sanger just a few weeks ago, and I was impressed how basically the underground car park was not accessible converted to a sample sort of samples in sort of station, and very typical of how Sanger approaches things. But the first question that then occurs to me, and I can ask that because I've been part of this, is where you're simply just sequencing the hell out of it and sequencing every single sample you could possibly get your hand on, or was there some actual power calculations as to how many samples you should be sequencing to follow this?
There was we were basically doing the first. There was various times at which this developed that we thought, well, this is the right number to do, and I think actually after the alpha wave, the British government just decided, well, we got this, the capacity going so high and the cost per sample of sequencing ended up being so low that we'll just do as many as we can kind of fit through. And that is overkill. I mean, I think you could easily make that argument. But it did, those last examples where you get I think the UK became this sort of reference point to get very fast response on things like vaccine efficacy against a new variant, because we would have a big wave and suddenly have all of the samples sequenced, and so we could do the analysis. So I think there was value in it, even though I think it's a fair criticism that it's you could call it overkill.
And I suppose the sample flow is not much reduced, right? Because there's probably less testing going on.
Well, exactly. So I sort of stepped down at the beginning of this year from an active role. And yeah, the testing flow is going to go way down. I assume they will keep sequencing some of it, but it's not going to be at that same level because there just won't be the tests.
Yeah, and that's of course an interesting question that takes us to the broader picture of how much what are the advantages of pathogen surveillance using whole genome sequencing, and how does one do it globally versus nationally, and at which level does one need to operate to pick up and monitor this particular pathogen, and how much should we broaden this out to other pathogens if we think about data-driven infection research now? So that was a long list of questions right there that might be of interest to discuss with this entire panel of speakers now. And so perhaps we can discuss these things and go around the table. But then, of course, other themes emerged here, right? Like how do we go from discovering a sequence variant to studying function and actually understanding the biology, and do that at the same rate as we detect these variants? And that is something that Jay and Laura brought up. And then the third point that struck me from all the presentations here, especially from Laura's, is of course the enormous capability that we now have to embrace biological diversity of not just pathogens, but also of the microbiome, and all these things strike me as potentially paradigm shifting. So we should be thinking, I think, if we build a community for data-driven infection research now, not just what the new opportunities are for and what we need to be doing, but also how that might change the way how we do things and how we collaborate. And perhaps converting a car park is just one example of how we need to change in times of a pandemic. But there are probably many other ways in which we need to change the way we operate. So perhaps we can discuss all these things a little bit and just go around the table. And so perhaps just returning to you, Jeff, because I already started with that first question of how to do surveillance in a pandemic. What is it that you have learned regarding pathogen surveillance and some of the points I raised earlier?
So I think probably the most important thing I've learned, which maybe other people who've done this in other contexts for a long time already know, is that all the hard problems are basically logistics. It's about, if you can easily imagine what sounds on paper like a very good design for how you're going to survey for existing transmitting pathogens, for emerging ones. but it's all about, can you actually just make that happen every day? And that is the thing we ended up building, was just a really smooth logistics machine in one place, in one country. It's absolutely the case that, 100 extra sequences from any other country in the world would have been worth a lot more than 10,000 more on any given day in the UK. It's just, you can't, the sequencing part of it really isn't that hard. It's, it's all the other components that go into it. So I think, and I know many people are thinking about this now, but the future thing, which I think should be, not just COVID focused, but as best as possible deployed to a wider range of, well, maybe first respiratory viruses, but then, in many other use cases, can you take any of the stuff that we've built and basically say, we're going to keep running it at some level, but collecting from a wider range of, of samples, such that they're not all just COVID, or they're not all just concentrated in countries that happen to have humongous existing genome infrastructures.
Can I just come in there? And we discussed this many years ago, also with respect to malaria and global surveillance there, that we should really send out sequences to all endemic countries and bring samples back. And perhaps that also resonates with Jay, who is studying and working in many African countries, right? Do you think that is a realistic way of doing it, or do we need or do we need to bring the samples to the sequencers, as you did in this case? And you argued for that, right, as being very efficient, Jeff? I don't know, perhaps Jay has a thought on this as well.
Well, I think we need to, I think in a way we're at a wonderful moment where we don't have to do all the sequencing in just one or two centers around the world. I think we need to be looking towards capacity building in low to middle income countries, particularly as diseases are causing big problems in particular countries. And we're now instigating different therapeutic options and different vaccine options. So really, not only do we need to be sequencing certainly in regions of Africa, if not within countries of Africa, we also need to be doing it prospectively. We need to know which variants are causing disease now, which ones caused a problem a decade ago, and which ones are going to be a problem in a decade's time, perhaps when new antibiotics, new therapeutics, or new vaccine programs have been rolled out. We need the backstory as well as what's coming next.
Yeah, Laura, any thoughts on that? And what was discussed before?
No, I think it was, so for me, it's because Jay did an awesome job touching on this. Sequencing inequalities and inequities are just in my research, a huge issue. I do some bantering with Salmonella, and if you search in Terrobase for Salmonella genome sequenced in Africa, I think I looked last night, it was less than 25%, I think, come from Africa, despite the fact that the highest burden of salmonellosis is in Africa. The vast majority of genomes are from North America, primarily the US and Europe. And sequencing small sets of isolates in under sequenced regions, you can get so much information about that, or about a pathogen's evolution, just even starting things there. So I think that's something that I think that's going to be a huge thing for any disease. It's so inequitable right now in terms of its distribution.
And then that also applies to the capacity and resources to analyze data, right? Equity also needs to encompass that. So if we want to have truly data-driven infection surveillance around the world, we also need to empower all countries to be able to do that and benefit from the data that are made available. How can we achieve that?
Well, I think there are many good open source pipelines available now that weren't available, say, 5 years ago. And most computational biologists have a really big open source philosophy, which does allow people in other countries to use the data and to process the data themselves. In terms of whether they need really big computers to do it, I mean, I'm very lucky to be sitting next to a very big cluster with a huge number of cores, so we have no problem with the computational power. How much of it can be done just on a laptop, I don't really know.
And then another, metadata is another question that was raised. And Jeff, do you have metadata? Do you get your virus data with disease outcome and that sort of thing. That would be cool to have, right? Or whether the person was immunized or not.
It would be very cool. We got very little metadata, just basically when the sample was taken and where it was taken, which is enough to do some kind of interesting epidemiology. And the linkage to, and the reason for that is, you know, these are samples taken without specific consent for research, you know, under a sort of public health exception. So it's totally reasonable that individuals' privacy has to be protected. So all of the linked analysis that led to, for example, the vaccine efficacy and hospitalization analyses, those are done, you know, sort of within UKHSA, who do have the fully linked data sets. That sort of is, I think, entirely reasonable. I think it would be great to get, you know, more of those metadata as anonymized as possible, made available to a wider community of researchers, because I think there are lots of interesting questions, and, you know, the investment to generate this genomic data set can best be leveraged if, you know, the research community has access to kind of the richest possible metadata around it.
Yeah, that is
the approach we took, Oliver, was we told people in different reference labs around the world, if they wanted their samples sequenced, we needed metadata, and they had to send us the metadata before they sent the samples. So we know the age of the patient, sex of the patient, what type of ice, is it from stool, is it from blood, etc. And exactly, normally we know cities and countries. We don't have any finer grain details. We'd love to know outcomes, and that is more difficult. And of course, we also don't know HIV status, because that is confidential, appropriately confidential patient information. So of course, we'd love to have more metadata.
Yeah. But these are important lessons, I think, for us to learn when we think about what sort of computational infrastructures we want to support as well. So we need to make sure we get the metadata, as much metadata in, and that we operate globally and are internationally connected in whatever we do, I think.
What I've realized is that the genomes get released quite quickly, but the metadata gets released later when work is published. And so there is actually a shortage still of good metadata, and lots of our metadata isn't in Enterobase yet, Laura, for instance.
Yeah, and there was a lot of, I don't know if this is relevant to you guys, but there was a lot of effort that had to go into even to get those internally linked data sets when you kind of think it should all just work. But of course, there's 62 different, you know, public health databases that all have to be made to work together, and huge amounts of effort had to be put into making that work. And it sort of does work much better now, I think. So I guess the hope is that within the UK, within other countries that are doing this, and indeed across countries, you know, we can learn some of those lessons to make it actually easier the next time, rather than starting from scratch again.
Mm-hmm. Laura, I just want to quickly touch on one aspect before we need to take a break, and that is, of course, how you tapped into the enormous diversity of microbial genomes, and so you set up a bunch of collaborators to then do functional follow-up studies. I think we can now, and Jay's work has shown that, right? We can use transcriptomics, for instance. We can use generic approaches to assess function of genes. If we can perhaps culture organisms, but perhaps we can take this to the single cell level and then deconvolute even complex communities of microbes and so on. But what do you think? Would you like to see? How should we operate differently to allow you to get at this biodiversity and at the function of all these genes that are out there?
Oh gosh, that's a good question. Well, so I guess so for me, it's like, okay, I find this cool thing, I give it to someone else, hopefully they think it's cool. But right now I'm trying to work on developing additional post hoc methods that allow people to kind of prioritize BGCs that are interesting in my specific case. So for example, this one is predicted to have antimicrobial activity. So I trained a classifier to do that. But in that sort of situation, it didn't turn out that great. It wasn't very good because I didn't have access to a lot of this information, because in publicly available databases, this kind of data was sparse. So I guess for me, someone brought this up in a question that I never got to answer, but I think there's there can be definitely biases in your data. And overcoming those is going to be like a huge step in terms of like which organisms we're studying, which like, I guess like biosynthetic gene clusters we're looking at, or genomic elements of interest. And model-based approaches are great for trying to, I guess, extend a framework to, I guess, the whole microbiome world. But obviously you're still going to encounter biases in terms of which data you're collecting. So there has to be like more incentives for supporting, I guess, like database maintenance and like curation and data generation. I don't know if
okay, great. So that's something we'll need to talk about as we evolve our program. So there's one quick question from Sana in the chat, and she points to the challenge of then, once you have found an interesting variant using sequence-based approaches, that you actually need to get at the isolate if you want to do follow-up studies. So
can I answer that one? I mean, I've been struggling with that in the past. Some of the early genomes, we couldn't get the strains. So I now have 2,000 African Salmonella strains in my freezer in Liverpool. But the other thing I'm doing is I've just got permission from the Malawian government, who own these strains, to put 5 of them, the 5 most important ones, into the NCTC culture collection. So in the next couple of months, African Salmonella will be available in the culture collection. And we really hope that will encourage researchers to use them as model strains and build on the data we've generated.
Yeah. Well, lots of things to think about. Thank you, everybody, for especially our speakers, for joining this session and the discussion. We'll still have a breakout session where some of the audience and perhaps some of the speakers might still pop in at 4:30. So I'll invite you back, and then perhaps we have a sort of open microphone discussion of some of the issues and others that we just raised. So thank you very much for joining us today. And I think we should all have a coffee break now.